everybody. Thank you for joining. My name is Lena Thomas, and I'm the Program and Engagement Manager at C2ST. Welcome to today's program. This program is presented to you by the Chicago Council on Science and Technology in partnership with the Carl R. Woese Institute for Genomic Biology and the Chicago Engineers Foundation. It's also sponsored by the Kleinman Center for Genomics in Business and Society and the Walder Foundation. The Carl R. Woese Institute for Genomic Biology is where science meets society, an interdisciplinary institute dedicated to transformative research and technology in life sciences using team-based strategies to tackle grand societal challenges. IGB members conduct groundbreaking genomic research to address issues in the areas of food security, engineering, health, technology, and environmental conservation, with work ranging from basic research that expands the horizons of human knowledge to applied research that builds on the foundational knowledge to create new technologies. This program is also sponsored by the Don and Catherine Kleinman Center for Genomics in Business and Society. The Kleinman Center serves as a platform for enhancing and accelerating the broader impact of the Carl R. Woese Institute for Genomic Biology's research and innovation, building on IGB's innovative genomics research by enhancing its global influence. The center helps IGB provide unique opportunities for economic development, public engagement, and social impact. The Walder Foundation is a private family foundation based in the Chicago area that provides individuals and organizations with the resources and tools to create more welcoming and vibrant communities. And for those of you that are new to the Chicago Council on Science and Technology, C2ST is a 13-year-old not-for-profit organization that seeks to enhance the public's understanding and appreciation of science and technology and their impact on society. C2ST hosts about 30 free or low-cost programs throughout the year on any and all science and technology topics. Today's program is the 8th of C2ST's Science from the Frontline series, an ongoing series of virtual events tackling relevant topics to the COVID-19 pandemic. This series also serves as a replacement for our canceled gala and will fundraise for C2ST. Please visit c2st.org to learn about how you can donate to this effort and get information about our upcoming programs. Links will be posted in the comment section below. Now, before we begin, I want to remind everyone to type in c2st.cnf.io into your web browser and click today's session to ask questions throughout the talk and also evaluate the program at the end. Again, that's c2st.cnf.io. If it's easier for you, you can also use the comment section and our moderators will make sure that the questions get to us. Now, I'd like to begin by introducing today's speaker. Rashid Bashir is Dean of Grainger College of Engineering, the Granger Distinguished Chair in Engineering, and Professor of Bioengineering at the University of Illinois at Urbana-Champaign. His research interests are to develop new micro and nanotechnologies to address grand challenges at the interface of biology, medicine, and engineering. Without further ado, let's welcome Dean Bashir.
Thank you very much. I really appreciate this opportunity to speak today. And can you see my slides now? Okay, great. So I will talk to you today about, spend a little bit of time talking about the Grainger College of Engineering. I'll also introduce IGB and also then take this opportunity to talk about some of my research interests across the spectrum of engineering, biology, and medicine. So just very briefly, our Grainger College of Engineering at the campus of the University of Illinois at Urbana-Champaign is the second largest college in the country. And again, as I always say, our goal is not to be the largest or second largest, our goal is to be the best. We have now 15,000 students, about 10,000 undergraduates, and about 4,500 graduate students. We have about 550 faculty members, about 470 tenure track, and the rest teaching faculty. And we do about 240 million of annual research expenditures that are basically coming from NIH, NSF, and various other funding agencies and industry. We are really proud of our alumni and our students and our faculty, of course, and our staff. And again, at the end of the day, every organization is about people, and trying to attract the best and the brightest from around the world is one of our major goals. And we are proud to have alums that have literally gone and changed the world. And there's nothing today in the field of digital transformation, in the field of electronics, photonics, the internet, the computer industry that our alums haven't created or touched, ranging from transistor, LED, and so many other inventions, YouTube, PayPal, and so on and so forth. So again, if there's any one thing that keeps me up at night as the Dean of the Grainger College of Engineering, is that how can we not only continue, but accelerate that trajectory of excellence, scale, and impact. This past year was also a very special year for us. The Grainger Foundation, based in Chicago, actually have been tremendous supporters over the year. William Wallace Grainger graduated from our college 100 years ago. In 2019 was a 100th year anniversary of his graduation. And the foundation decided to give us another gift that added up to the largest amount ever to a public university college of engineering. So as of last year, our name is now the Grainger College of Engineering, and we are really very, very proud of that. I also want to give a big shout out to the Institute for Genomic Biology. It's the largest comprehensive genomic institute in the country on our campus, and I am myself personally a member of that. My research group benefits tremendously from collaborations and the very unique facilities and the excellent interdisciplinary convergent environment that exists at the IGB. And it's a hallmark of the collaborations across our campus. So this institute, again, is co-sponsor of this series, has made tremendous impact on our campus and the world. Let me briefly talk about healthcare just as a broad challenge. And we know that in the United States, our cost of healthcare is really unsustainable. The rate of increase is unsustainable. Today, we spend about 20% of our GDP in the country in healthcare. This plot is a couple of years old, but the trends are still very valid. And we spend about 2x that of any other developed country. So this idea of this triple aim that's mentioned here, that how can we provide higher quality for more people at lower cost, meet all three goals, is called the triple aim in healthcare. And this is something that's just a grand challenge for all of us to really address. And what we really need is like a Moore's law for healthcare. So what electronics and computing has done for healthcare, where every generation is potentially cost per unit is actually cheaper, is available to more people, and it's at higher quality. So I think this century is just a century of convergence across engineering, biology, and medicine as the global population continues to increase, and especially in the less developed countries. People are living longer and want to live healthier life, which is great. In the United States and in other parts of the world, the number of elderly is increasing, and we need to do the best we can to make sure that they live a very happy and fulfilled life. So this convergence, as I mentioned, is really what we believe is the next frontier, and what can we do for medicine that engineering did for electronics and computing, very broadly speaking. So those are some of the grand challenges. And these vision and this goal is really what we truly believe in at engineering at Illinois. Of course, I put the College of Engineering here in the middle of the circle, but we truly also believe that these grand challenges can only be addressed via collaboration, like the one across engineering, Grainger Engineering, and IGB, to develop interdisciplinary solutions to improve human health and the human condition. Not just with biological sciences, but also with social sciences, with ag, physical sciences, of course, as what engineering is really derived from, and also business law. So we have lots of collaborations across our campus and beyond in these areas. So I also want to just point out this one article that myself and a few other leaders in the field had written a few years ago in Science, Translation, Medicine, with this idea that the integration of engineering and medicine into engineering until these boundaries vanish. This is really going to be crucial, especially in the coming years, not only in the current COVID crisis, but also in general in the future, to really be working in this different fields in a very convergent manner to address these grand challenges. And then there's also this really great report on convergence, future of health from MIT that I would encourage you to look at. So along this line, I want to take this moment also to remind us that our campus is the home to the nation's, as a matter of fact, the world's first med school focused from the beginning on the intersection of engineering and medicine. This is something I was personally involved in helping to get started. So the idea that with a simple hypothesis that just like you can't be an engineer without knowing math, we believe that in the future, you can't be a physician or a physician innovator without knowing engineering and technology. And if that's the case, then why don't we integrate these concepts into the curriculum, into the medical curriculum itself? So we started this college a few years ago. Our third class is due to start this summer in 2020. And the curriculum includes engineering, medicine, biology, and medical humanities integrated all across the 4-year. And we're really very, very proud of being the first to do this. And this is really proceeding very, very well. I want to also talk about this one other center that we have that's led by the Grainger College of Engineering through our Healthcare Engineering System Center. And this is in partnership with our other close clinical collaborator, which is OSF Healthcare. So this partnership is called Jump Arches, and it's backed now with a large endowment where the income from that endowment is used to do clinical translational projects across OSF Hospital in Peoria. They also have a presence now in Chicago, and working with engineers and social scientists at the campus, at the Urbana-Champaign campus. So the idea is to connect engineers and physicians, and also bring in social scientists and behavioral scientists to look at the social and behavioral determinants of health. So this is a very exciting center that I encourage you to look at the website and learn more about. Another thing that I want to talk about just very briefly, and it's become even more so important just with all the events in the last month or so, is actually early in the spring, we started a new institute in the College of Engineering called the IDEA Institute, which stands for Inclusion, Diversity, Equity, and Access. And it's a novel concept for a College of Engineering to have an institute within it. It's led by one of our star faculty, and the idea is to support scholarship, innovation, collaboration, and leadership in the areas of diversity, equity, inclusion. And this has become even more important in the last month. And if you go to the center, if you go to this institute website, you'll find Zoom recordings of multiple conversations that we have already had on our campus that this institute led to discuss these critical matters of racial inequality and social justice. So this institute has played a critical role in these conversations just in the last month, and hopefully will advance our efforts in these areas. And then, of course, we wouldn't be where we are unless we certainly talk about COVID-19 and this pandemic that has hit us all. These are numbers as of yesterday. I find myself still going and looking at these plots every day, at least once a day. As you know, the numbers are increasing again in many states across the US. But what I want to really just focus on is the areas of impact that our campus has really stood up and responded. And I've just been so humbled and amazed at our faculty colleagues and the whole campus, our staff, our students who have stepped up to respond to this challenge. And I'll just mention a few, but these are all the things that have been going on that our campus is leading. We have faculty who are working on epidemiological modeling of the disease spread and advising the governor's office, Governor Pritzker office. We are working on expanding diagnostics, and I'll mention that in just a little bit. Increasing ventilator capacity, and also various mitigation approaches for airborne transmission, developing new PPE devices and so many other things that we have really, our campus has stepped up to respond to this. So just mention within a matter of, this happened basically the third week of March when the pandemic had just taken off, working with Carle Hospital in our town. We stood up a team led by Professor Bill King in mechanical engineering, a group of about 40 people that stepped up and basically designed an emergency ventilator within a matter of about a week or so, and then developed a prototype and built it, and it worked, and then did animal testing. And eventually this design now is being commercialized. This was actually licensed out to about 50 different entities around the world. And the license was then also taken up by Foxconn Interconnect Technology and their subsidiary Belkin, and they are now actually planning to commercially produce it. So you can find news in all of this, but this was just an amazing example of impact that our faculty and staff made shortly after everything happened with COVID-19. It's called the Illinois Rapid Vent, and you'll find a lot more information on our website. Similarly, our campus right now, I'll also just give a quick shout out, has a major effort around target, test, and tell. It's called the SHIELD team, led by Professor Marty Burke from chemistry and from the Carle Illinois College of Medicine. And again, this is a campus-wide effort which is working on trying to, scientifically and data-driven, data-informed way of targeting algorithms for trying to prioritize testing, and then developing a new saliva-based test for COVID-19 that is fast and very scalable. And as a matter of fact, it has been made available this week to our campus faculty, staff, and students that are here for free. And this is a critical part of our fall opening plans for our campus. And then finally, being able to then notify whoever chooses to download this Rockwire app, which has been developed at our campus, using this to then do contact tracing of the individuals that were tested. So this complete comprehensive approach is going to be critical to making sure that everyone is healthy and safe. So with that, I'll actually just kind of switch gears and tell you about a couple of projects that I've personally been involved in the next 5 minutes or so, and then I think we should still have plenty of time for questions and answers across anything that the attendees would like to ask. So my group is also very interested in developing personalized approaches for infection and sepsis, and this, of course, comes in very timely with everything that's been going on. But the idea of being able to take body fluids, whether it's saliva, sputum, urine, venous blood, or a finger prick, and then performing detection of the pathogens, trying to see if we can do specific therapeutic and diagnostic therapeutic management using the pathogens that have been extracted. If you can isolate those pathogens, then you would like to do drug susceptibility testing and study. And then also potentially looking at the profiling of the immune system of the individual, because everybody's immune system is different and different people will respond differently to infections. So not only the detection of pathogen is important, but also trying to figure out how the immune system is doing for that individual is also very important. And that can be done by measuring specific cells, proteins, or DNA. And then again, connecting that back to the appropriate therapeutic management. And then also taking all this data that is obtained by doing this kind of measurements and then using data analytics and machine learning to then really gain additional information so that you can build a database. And then, for example, you build a database, let's say with 5,000 patients. And then when the 5,001 patient comes, you can learn from the previous 5,000 to be able to predict and stratify the future disease state for that next patient. And then you kind of keep building that. So this has become a really important element of this idea of personalized medicine. Okay. So we started a project some years ago on sepsis and precision medicine. And we pulled a team together that included people from my group that are working on the technology and sensors devices, but then the clinical partners from Carl Foundation Hospital, biologists from our campus, and some machine learning and statistics faculty from across our campus to really pull this very interdisciplinary team together. And the idea has been to look at sepsis in this way where you measure certain biomarkers from the patients, and then use the electronic medical record data for the patient and those biomarkers that are measured to then develop a machine learning model, and then use predictive analytics to then stratify, like I said earlier, the patients that would come next. And then in parallel, also develop a point of care device, which is shown here on the right. This is a credit card size microfluidic device where you can put a drop of blood and count specific cell types that indicate the progression of sepsis, for example. So doing all of this in parallel, we are able to improve the predictive accuracy to help the physicians. So what this curve on the top right shows is what's called the ROC curves, which essentially tells you the predictive power of the assay. And this orange line and this purple line actually is from the current clinical practice through what's called a SOFA score. And as you can see, this is in the 0.4 to 0.5 range, which really means that today physicians are looking at a couple of criteria when they predict a patient is going to get sepsis or not. And it's like a coin toss. It's really 50%, essentially. They can be right or they can be wrong 50% of the time. Whereas if you start to add our biomarkers and the biomarkers that we were able to find and measure, you can increase their predictive accuracy quite significantly. And this is actually a little bit older data. Now we are up at over 0.9 with this sort of measurement. So this work actually we also have translated into a company called Venosus, which is now based in Chicago. And they are working on building the largest sepsis database in the world to be able to stratify patients at a hospital. They've raised some funding and have multiple hospital partnerships. So it's really very exciting from that perspective. I'll also mention from our group, we've been also very interested and been working on detection of viruses on a chip. So again, this it's been a very busy few months for me personally. Of course, working to lead the college, but also I just couldn't stay back from a research perspective because my research is exactly in this area of viral detection. But we'd already worked on demonstrating HIV, HCV, Zika, dengue, and chikungunya on a point-of-care device. And this is in collaboration with Professor Brian Cunningham and Professor Bill King from our college, where we develop a microfluidic device. You can put a drop of blood, it gets processed in this cartridge, and then you can look for specific pathogens. And again, we demonstrated these different viruses on a chip. And then the amplification of the RNA from these viruses, or essentially the detection of these viruses, can then be performed using a cell phone camera that is connected or sort of inserted into this cradle, into this small portable cradle. So this is work that we were already doing, and now we are working on translating this approach to detection of coronavirus from nasal swabs and saliva. This is in collaboration with OSF, but also a very active collaboration with Coral Hospital here in town, where the goal is to be able to either, let's say, take nasal swabs, but really more and more, we believe the present and the future is with saliva testing, and be able to then do this isothermal assay development that our group has been doing, and then develop an instrumentation for development for rapid detection. So we've gotten some initial results on this that are on a bioarchive and also been submitted to a journal for publication. Here's 10 samples of patient VTM that we got from OSF Hospital in Peoria. And you can see that the positive samples that they told us were positive, we also find them to be positive from our technique. And the negative samples, these one in red that were confirmed negative also, we don't find any amplification from those samples. And these curves right here kind of show that pictorially. These are the positives where these fluorescence curves go up, and the negatives are where the fluorescence stays very, very low. And then we have also demonstrated the detection of these coronaviruses from the viral transport media from the nasal swab in a portable device and cartridge, like I showed earlier. These are new additively manufactured cartridge that we can inject the sample and the reagents, and the reaction happens in these wells right here that is then detected by the cell phone camera. So using that also, these are again initial results, but very promising, where we can detect the presence of these viruses within a matter of 20 to 30 minutes, let's say 30 minutes, but the results here are shown within 40 minutes. You get very clear distinction between the controls and the positives. So in the last couple of minutes, I'll just mention something else beside COVID. Of course, COVID-19 has taken over our world, and it should, but we have other things also that are going on within our group. And I'll just very briefly mention this idea of building with biology and the idea of engineered living systems. And this is something that, again, is a team in the Institute for Genomic Biology that has been started just a few months ago. But we have been working on this research for the last 10 years, actually, as a matter of fact, with many colleagues from Illinois and in a collaborative center funded by NSF with MIT and Georgia Tech. And now, as I mentioned, this work is now continuing on as a team within the Institute for Genomic Biology. And the idea here is can we build engineered living systems? Engineers are used to building devices in silicon and plastic and metals, as I just showed these microfluidic devices. but can we learn the design rules of building with cells? And could we engineer tissue-like structure that can do certain functions? And I'll just show you some results just to give you the idea. So here is what you'll see here, if you can see this video. This is a 5 to 7 millimeter long structure that is what we call the biobot, a biological robot. It has this scaffold, which is a 3D printed hydrogel structure. But then this layer that's in white, kind of whitish color, this is actually a skeletal muscle from mouse that are responsive to light. So we have optogenetically transfected these muscles. And when you shine light, the muscle actuates. And as the muscle actuates, it actually moves the small biological robot across the petri dish from left to right. So this can all be forward designed, essentially, by 3D printing and by tissue engineering and integrating it together to form these biological robots that can, in fluid, do some work and provide this actuation. And again, we can characterize it and do all sorts of design. As a matter of fact, now we have an undergraduate bioengineering lab that uses these protocols to teach students the sort of the basic design rules. And then students can design any kind of structure that they wish, pumps and muscle rings and valves and all sorts of biomechanical structures. Similarly, this is just another example of a device that has 2 muscle rings, and it can move to the right or the left depending on how you actuate it. Here is one that can actually turn also depending on how you actuate it with light. So again, as an example of this, so biological fabrication with cells and muscles. So with that, I'll actually just sort of end the talk and just talk about just briefly the excitement around the convergent research with engineering, biology, and medicine, and lots of opportunities to really make an impact in healthcare, from diagnostics, which is really very relevant nowadays, of course, and will be in the future, all the way to tissue engineering. And then broadly speaking, how we want to bring together our faculty from College of Engineering, the Grainger College of Engineering, with researchers across IGB and across the campus and beyond to solve these important problems. Thank you very much.
Thank you, Dean Bashir. We have a number of questions in already, but I want to remind everyone to go to c2st.cnf.io to send in your questions, or feel free to use the comment section below. I'm going to start us off with our COVID-19 focus questions, because there's quite a few. So to start us off, what long-term effects do you think COVID-19 will have on your field?
That's a great question, and it's a very big question. Broadly speaking, I mean, COVID-19, I think, has obviously changed our world, changed the world of higher education, and changed the world of research in many, many ways. And I think the new normal won't be where we were. So I'll just try to address a few things. Certainly, you know, in the field, in the world of higher ed, I think being able to do online and how much we should be teaching online, how much we have to teach online versus how much we should be teaching online is a big question and a big debate. What's the best mode of learning by combining hybrid models? What's the best way to assess in these remote environments? Those are all major questions that we have been working towards. I think in terms of research, clearly, you know, addressing a range of challenges that have come about due to COVID-19 pandemic, from diagnostics, rapid diagnostics, personalized diagnostics at home, all the way to developing ways to rapidly develop drugs, to develop ways of communicating with each other that are safe and private, all the way to what the future will look like with you know, being ready for other pandemics in the future. So it has really, you know, changed the world for everybody and for all of us in all of these various fields.
Okay, thank you. And with the virus detection technology you're working on for COVID-19 let me say that again. Would the virus detection technology you're working on work for COVID-19 antibody testing?
Yes, that's a great question. So my group itself is actually not working on antibody testing, but I have other colleagues on campus. We will have a test on campus actually for antibody testing that we want to be able to, you know, test our students and faculty and staff in preparation for fall opening. And I also have other colleagues from our college that are working on point of care rapid tests for the antibodies. And this is something really important, as you know. And the fact of the matter is, we're still learning. We're still learning about COVID-19. We do not know how long the antibodies stay in a person in the blood when one is infected. As a matter of fact, as of now, I think if you look at the literature, no one that we know of has been reinfected. When someone is infected, they get better, we don't know of any cases where they have been reinfected. So we don't know how long that would be the case. Perhaps after some time that immunity will go away, but we don't really know. So that test is really important. And there are rapid tests that have been developed now and are continuing to be developed that would allow one to test antibodies very quickly from within your blood.
And is the goal to make those technologies, particularly cell phone technologies, is the goal to make it available to the broader public? And if so, how does it affect the roles and relationships between doctors and patients, in particular, how doctors are easily dismissing patients for doing their own research for Googling their symptoms?
Yeah, that's a great question. And that's really a broad question, I think, just around what the future of medicine looks like. There's no question that, digitalization of medicine is happening and should happen at a faster rate, and it will continue to happen. So there's no doubt that patients know so much more now than they did before, and I do believe that more and more, pre-COVID-19 and certainly with COVID-19, many aspects of primary care actually, will become more and more digital. In the sense that, having the patient, rather than the patient going to the lab will start to come to the patient in the form of these point of care diagnostics. So this is something that I believe is going to happen, and it's probably happening at a much more accelerated rate because of COVID-19.
And then back to the ventilators you briefly mentioned, how do they differ from existing ventilators?
That's a great question also. So the ventilator that our team had designed from our College of Engineering is actually an emergency ventilator. It's a portable device that's less than $200, and it works by having it plugged into the oxygen supply from the hospital room, or it could be a portable tank of oxygen. And it works actually for, let's say, 5 to 7 days, and then you throw it away. So this is an emergency ventilator. There is multiple designs of this kind of devices out in the market, but really there was a supply chain issue. These devices weren't available when they were needed back in April or so, let's say, in New York. So the traditional designs are many thousands of dollars, and they're much more sort of sophisticated and advanced. But however, our design is portable, handheld, and meant to be used for a short period of time, like I said, 5 to 7 days, and then disposable, and then you put a new device basically.
Okay. And I'm going to try and combine these two questions because I think they're very related. Do you have any thoughts on aerosol transmission of coronavirus inside and outside? And could the ionization destroy the coronavirus? And if so, should HVAC systems be added to schools, businesses, and hospitals?
So that's a great question. We actually have some of the world's experts in this area in our college. I would encourage people who are asking the question to send me an email, rbashir@illinois.edu, and I can connect you with folks. As a matter of fact, on our website, there's information on projects in this area. We have faculty that are studying that and looking for ways to sterilize, for example, PPE and masks to be able to reuse them, and use other methods of, for example, ozone-based methods to disinfect. So yeah, there are methods like that. And again, one very important point there is that even though there's many methods being developed, at the end of the day, they have to go through some sort of an approval process with CDC and FDA, depending on the application, to actually be available for broader use. So we need to always make sure that the methods that are being developed are safe for people to use and are approved appropriately.
I think you briefly mentioned this for the UI/UC campus, but should antibody testing be used to determine the safety of students and teachers returning to face-to-face learning in the fall?
Yeah, that's a fantastic question. So yeah, our campus has a very forward-looking and a very comprehensive approach. We believe it's really very unique to our campus that this effort has been started. As I mentioned, it's called a SHIELD. Professor Marty Burke from chemistry and the Carolina College of Medicine is leading it, but it's a large team of faculty and staff from across and clinical partners and the Champaign-Urbana Public Health District, now working also with IDPH in Chicago and the state. So point being, there's two types of tests you like to know. First of all, of course, the diagnostic itself, looking for the virus. And that, again, our approach is to go with a saliva-based test, because we believe that that's really, of course, so much easier to collect, not as invasive as the nasopharyngeal swab, and also very scalable. So, and there is multiple research studies now, including some of our own, that has shown the equivalency of the saliva test to the nasopharyngeal test, a swab test for the viral detection. So that's one piece. You want to make sure that whether somebody's infected or not, of course. And then the other piece is that that immunity piece with the antibodies. That's the second piece. So our approach is to be able to do both of them on our campus as we welcome students back for fall. Our campus, we are actually setting up to be able to do up to 10,000 tests per day, 10,000 saliva-based tests per day on our campus.
Amazing. Now we have some really great broader questions that I think will touch on the interdisciplinary part of your work. How can engineering help bring down medical costs down? How can engineering help bring medical costs down for future generations?
Well, that's an excellent question. Of course, we can have a whole entire talk on that. So I think there is, of course, many opportunities, and where engineering approaches can help reduce cost in medicine. And it can come from, first of all, just improving processes and efficiencies. So there's lots of processes and lots of efficiencies that can be gained within hospital systems, within the whole medical enterprise. And of course, it's a very complicated, complex system of healthcare providers, insurance companies, reimbursements, and all. Of course, coupled with policies and other intricacies. But the bottom line being that I think the idea of being able to forward design systems that improve efficiency, eliminate errors and waste, there's a lot of opportunities within the whole healthcare system. A very large percent of the sort of costs of healthcare are attributed to, for example, medical errors, where we believe that technologies can be used to aid physicians and healthcare providers avoid those. Secondly, there is a lot of instrumentation and sensing and diagnostics that can be done that is also lower cost. And I think the challenge in that space is this idea of connecting very high precision, high cost, personalized diagnostics. That's really important, of course. Connecting that or bridging that gap from that to population health, and thinking more and more about prevention and wellness. So how can we detect disease at a much earlier stage? And then how can we do population-based studies to be able to predict and stratify patients' healthcare? So I think connecting technology and engineering with data sciences, and then also connecting it with social and behavioral determinants of health. So really bringing all those aspects together, I think in a very convergent way, is really the way to go in the future to reduce costs of healthcare.
Thank you. And we have our final question for the day, which I think is very related to what you just said. How do you see medicine evolving in the future, and what does your ideal version of it look like?
So like I said, I personally believe, and I know many of my colleagues on our campus certainly share that, and across the country, that I think just like, as I said earlier, just like you can't really be an engineer without knowing physics and math, I believe that in the future, all physicians would need to know technology, would need to know some aspects of engineering, technology, and data sciences and computing, and be the drivers of the change. So I think the future of medicine is going to be very digital. And of course, while doing that, we must make sure that we don't lose the human aspects of medicine. And as a matter of fact, the goal when we talk about integrating technology into medicine, the goal is to allow the physician to be more responsive to the human aspect of medicine and not to be spending as much time, so to speak, on the other aspects of it. Similarly, the other sort of broader goal of using data sciences and technology is to learn, have the future physicians learn from the experience of the past, but not just from one person who trains them, but rather the collective experience of thousands and thousands of physicians. So the idea that if I can capture the experience digitally with the patient medical records and what the treatments that are being prescribed, and if I capture all that and develop machine learning models to learn from that, to be able to predict and treat and stratify the next patient, I think we can only make the entire process so much more efficient and higher quality and hopefully lower cost, while maintaining that human connection that is critical in the field of medicine.
All right, thank you so much, Dean Bashir. I want to thank everyone for tuning into today's program. Please remember to visit c2st.cnf.io to evaluate today's program for a chance to win an Amazon gift card. Thank you again for Dean Bashir for taking the time to join us today, and thank you to our partners at IGB, the Chicago Engineers Foundation, the Kleinman Center for Genomics in Business and Society, and the Walder Foundation. If you enjoyed today's program, please consider donating to our fundraiser. See the pin link in the comment section below, or visit c2st.org to find out how you can support this virtual series and the fight against COVID-19. Thank you again for watching.
Thank you.